Embryogenesis is the process of embryo development within organisms, and it is, as one would expect, quite complicated. In this video, I will be specifically going over neurulation and specification of neural crest cells. There are many steps involved, so if you ever feel lost, feel free to rewind to earlier parts of the video. I will leave timestamps below. With that being said, let's begin. Neurulation is the process where the three ectodermal regions, the epidermis, trunk and spinal cord, and head and brain, become physically and functionally distinct from one another. In other words, this is the stage where the vertebrae of an organism begins to form. It begins at the head region, or anterior end, as some will expect, and gradually progresses to the tail, or posterior end. I will be going over primary neurulation, which is present in the chick and mammalian embryos. We begin the elongation of the neural plate as cell division extends it along the anterior-posterior axis. Then, above the nodal cord and floor plate, a medial hinge point, or MHP, is formed, and like the name suggests, it acts as a hinge point for the neural plates to start bending and folding inwards, creating a neural groove. These neural folds then converge through dorsolateral hinge points, or DLHP, to close in on a neural groove. Lastly, when the paired neural folds contact across the top of the dorsal midline, the tube closes as the folds fuse with each other, leaving the overlying epidermal ectoderm above them. The cells at the top of the neural tube become neural crest cells, which we'll come back to later. But what are the morphogens responsible for this? First, a very creatively named gene called the sonic hedgehog gene is expressed ventrally in the nodal cord. The sonic hedgehog gene also acts as an inhibitor for the noggin gene, which is expressed in the dorsal neural plate. The noggin is a BMP inhibitor, which, inhibits BMP. And BMP, in turn, inhibits both the formation of the DLHP and MHP. All of these morphogens are kept at a perfect balance in order for neurulation to take place properly. For example, if the nodal cord wasn't developed properly or missing, then the sonic hedgehog gene will not be present. This means that there will be more noggin since there's nothing inhibiting it. More noggin leads to less BMP, as more BMP is being inhibited by the noggin gene, and with less BMP, the formation of DLHP and MHP will be exaggerated since there isn't much inhibiting them from forming. This will cause the neural tube to bend too much. We need just the right amount of morphogen for proper neural tube formation. My prof calls it the Goldilocks zone. So what about the neural crest cells? These cells become specified at the border of the neural plate and migrate away to the rest of the body out of ectodermal tissues. But how do they know whether they are neural crest cell? The answer is morphogen gradients, specifically BMP and WNT gradients. Taking a cross-section of the neural plate, this is what we see, with the nodal cord at the bottom here, dorsal ventral axis along here, and anterior posterior axis coming out and into the screen here. Remember from before, noggin, a BMP inhibitor, is concentrated at the dorsal regions of the neural plate. This means that there is a gradient of BMP inhibitors starting from the dorsal side and decreasing towards the ventral side. This means that the BMP gradient will be opposite to its inhibitors gradient, from the ventral to dorsal side. This concept applies to WNT inhibitors as well. WNT inhibitors, such as Cerberus and Dickoff genes, are concentrated at the anterior end of the embryo, which forms a gradient from the anterior to posterior end. The WNT gradient will then be from the posterior to the anterior end. This is easier to visualize if I put a face on it. Let's take a piggy for example. Its face is anterior and dorsal end of its body, so all of the gradients will look something like this. Shout out to one of my classmates for suggesting this. So why do we care so much about the gradients? Well, it is observed that where WNT and BMP signals are the highest, so posterior and lateral region, epidermis cells are specified. Where BMP signal is blocked, neural cells are specified, as mentioned before in the process of neurulation. Where WNT signals are followed by BMP signals, but then downgraded, posodal cells are formed. They are thickenings of the epidermis that will give rise to the eyelids and other sensory structures. And last but not least, where WNT signals are followed by BMP signals but remains active, neural crest cells are specified. But how do these neural crest cells migrate? I'll be doing another video on it, as this video is already long enough, and I also need to finish studying for the final for this class. So yeah, thank you for watching.